Hello there, welcome to Fundamentals of Biology, where today we're going to be looking at nervous systems. So during this session, we're going to have a look at diversity in nervous systems, so different types of nervous system in different animals. Then we're going to focus specifically on the vertebrate nervous system. And then we're going to go on to talk about action potentials, which is how nervous systems actually function. And then we'll go on to look at some of the main sensory systems that we see within animals. To start off then, when did nervous systems first become a thing within animals? Well, by the time we get to the Cambrian explosion, so this is more than 500 million years ago, and we know this from the fossil record, we start to see specialized systems of neurons. So neurons are one of the main types of nerve cells that we talked about before in the session on animal tissues. So we know that nervous systems are made up of neurons and supporting cells, such as glial cells. Well, we start to see these specialized systems of neurons within the Cambrian explosion. And it appears that they enabled the animals to sense the environment around them and respond really quickly to the environment. And it's believed now that this led to a kind of arms race. So you have an arms race between predator and prey and host and parasite, because now you've got animals that are able to sense the environment, they're able to respond rapidly to it, and that means that other animals need to evolve accordingly. So from that point on, we start to see a huge diversity in nervous systems, which are still evident today. So when we look at extant animals, so those are animals that are alive today, we see a really huge diversity in different types of nervous systems, from the very simple to the quite complex. So nervous systems range then in complexity from simple nerve nets to central nervous systems having complex brains and central nerve cords. So let's unpick that statement a little bit. So nervous systems range in complexity from simple nerve nets. Well, if we look up here, we can see that the simplest animals that have a proper nervous system are the Cnidarians. So you're looking at things like jellyfish and anemones and corals and this Hydra here. And what we can see is that they have a simple nerve net. And this is just a diffused network of neurons that spread throughout the body. And as we start to get into more complex animals, if you like, we start to see more complex nervous systems. So if we look to the next one here, where we look at an echinoderm or a sea star, we can see that they have a radial nerve. So a nerve is a bundle of neurons, specifically the axons, which we'll look at in a bit. So we see a nerve ring, and then you have radial nerves moving off and going into each arm. And what you can't see on this diagram is that coming off of each radial nerve would be nerve nets. So you'd have this diffuse network of neurons moving down throughout the body, but then they feed into these radial nerves that then feed back up to this this nerve ring. And that means that the sea stars or the echinoderms can actually coordinate movement, and they can coordinate senses, so they know what's happening in which direction. So that means that they can respond more actively. And then as we start to move towards more complex animals again, so here we have a Planarian. So this is a type of flatworm. What we can see here is the beginnings of bilateral symmetry. So we can see that this is an animal that has bilateral symmetry. And in animals, when we get bilateral symmetry, we tend to find something called cephalization. And cephalization is the clustering of sensory organs towards the head or towards the front of the body. And you tend to also then get the development of a brain. So when we look at these flatworms, so these very simple animals, we start to see bilateral symmetry, and now we've got cephalization. So they've got very crude eyes, so basically eye spots, which are just patches of light-sensitive cells around the head, where they also have a rudimentary brain. And then you can see that they have these nerve cords. So these are ventral nerve cords, so they run along the underside. So within vertebrates, the nerve cord runs dorsally, so like ours, down our back, and in invertebrates, it runs ventrally, so along their underside. And these guys have a twin nerve cord, and then they have these transverse nerves that feed between the two, so that enables coordination. And then as we move up again, so now we have the annelids, so Annelida, which are the segmented worms. So we've got an example of a leech here. Again, bilateral symmetry. We have cephalization, so we've got a brain. We'll have eye spots up here. And then what we're also seeing now, because we've got a segmented worm, you need to have some kind of coordination and control of the individual segments and coordination between the segments. So what we start to see now are things called ganglia, or segmental ganglia specifically. So you can see those here on the annelid. And what these ganglia do, they're basically a cluster of neurons, and they act as a relay center. So they take in information and they help to direct it into where it needs to go. So this helps to coordinate movement and information between the different segments. And we start to see these in more increasingly complex animals. So if we move over here to an arthropod, so we've got an insect here, we start to see, or we see again, a brain. We've got more complex sensory systems. So we've got the antenna, we have eyes, and we've got a slightly better developed brain. And then we've got a single ventral nerve cord. And then we've got segmental ganglia again. And then here we can see a chitin and a squid. So these are both mollusks, and I'm going to come back to these in a second. And then over here we can see a vertebrate. This in particular is a salamander, and then we can see here that we've got a pretty large brain in comparison to most of these, apart from maybe the squid. And then we've got ganglia again, and we've got a spinal cord. So this is a dorsal nerve cord now, so it's running along the back, which is unlike what we saw in the arthropod over here. This is much more like what you would expect to see within us, so it's quite a complex system. The reason I said I would come back to these two, so as we can see, the chitin and the squid are both mollusks, so they belong to the animal phylum Mollusca. But what we see with them is a brilliant example of the, within animals, nervous system organization and complexity will typically correlate with lifestyle. So what we mean by that is that, again, we've talked about this a lot, but it's very much a case of structure equals function. If an animal leads a complex life, and by complex, we mean that it is active, it is actively moving around, and it's specifically, if it is actively hunting, well, if you're going to do that, you need to have a more complex nervous system. So chitins are typically sessile, so they typically stay in one place or within one limited area, and they just sort of graze. They're not actively hunting other animals. Because of that, they don't have a particularly complex nervous system. Compare that to another mollusk, which is the squid. Well, squid are active hunters. They are actively searching for prey within the water, but they are also food items themselves. So they are also prey species themselves. So they are also trying to avoid predation themselves. So they are actively hunting other animals, and they are actively trying to avoid predators. And because of this, they've evolved to have quite complex nervous systems, interesting brains, interesting nervous systems to enable them to do the things that they do. Because if you think about it, actively hunting requires a lot of coordination between the different parts of the body. First, you need to identify the prey. So you need to find it. So that means you need senses, whether it's eyes or whether it's a sense of smell or a sense of taste, whatever it might be. You need to be able to sense and detect that prey. You then need to be able to coordinate your attack to try and catch that prey item. So you need to be able to approach it without scaring it off. And then you need to approach it in such a way that you can actually grab hold of it and grasp it. And again, this all requires coordination between the muscles of the body. Active hunting and actively moving around within your environment requires that you have a more complex nervous system. The more sessile you are, the more passive you are, So up here we've got the most passive of them all, all of these animals. We have a Hydra, which is a sessile animal, so it attaches by its foot, and then it just sort of moves around in the water column, filter feeding essentially, or trying to catch food within its tentacles. So not necessarily filter feeding, but trying to catch prey within its tentacles. But it's not actively hunting, it's a passive process. So this is what we see within the diversity of nervous systems. It's a very good thing to remember that the nervous system organization and complexity will typically correlate with the lifestyle. So when you look at an animal and you can try and identify the complexity of its lifestyle. So I don't just mean whether it's actively hunting and moving around, but also think about social systems. Does the species communicate? Are they learning? Are they passing on information? How do they interact with individuals within the same species? How do they interact with animals of other species? How do they exist within their environment? Are they manipulating or changing their environment? The more complex actions they do, the more complex their nervous system has to be to enable them to do that. So it's a good thing to always remember when you're looking at a species of animal and you're trying to work out what its anatomy and physiology is. So when we look specifically at the nervous system of vertebrates, as you'd probably imagine, we see quite a complex system. And that's because we live very complex lives, particularly obviously we can see here an example of the human nervous system. Well, it's about as complex as you can get, really. So vertebrate nervous systems, we separate into two main components, central and peripheral. So the central nervous system is composed of the brain and the spinal cord, obviously the brain up here in the head, and then you can see the spinal cord that comes down here that actually stops about 2/3 of the way down the back. These are the main processing parts of the body. Information comes into the central nervous system where it gets processed, or what we actually refer to as integrated, and then it sends out a response for the body to react. Okay, so this is where all of the computing power is, if you like. So that's the central nervous system. But we also have a peripheral nervous system, and this is composed of nerves and ganglia. So you can see all of the nerves, so the spinal nerves moving out here, and some of the largest cells within the animal body are found in vertebrates that go from the base of the spinal cord down through the legs. So you can see them moving down here. So these are essentially controlling homeostasis. So they are helping to control and monitor the inside environment of our body, but they are also detecting information around the body, so detecting sensory input from the environment around us, and then the peripheral nervous system will deliver that information into the central nervous system, where it will then get processed, and then the central nervous system will send a response out again via the peripheral nervous system. So if we look at that in diagram form, what we can see here, so along the bottom here we've got the peripheral nervous system here, central nervous system here. We have sensory systems that we're going to go on and talk about a bit later on. Sensory input comes in, it gets sent via the peripheral nervous system into the central nervous system where it gets processed or integrated. That will then determine what response you need, and that response is sent via motor output, again, via the peripheral nervous system to what we call effector cells. And those effector cells will be cells that are carrying out an action. So it will be things like a muscle cell contracting or an endocrine gland secreting a hormone. Okay, so sensory input comes in via the peripheral nervous system. The brain and the spinal cord will process that information and then send out an appropriate response, again via the peripheral nervous system, to effector cells that will basically carry out that response. What we also see about the peripheral nervous system, though, because there's so much going on there, it is monitoring and regulating our internal environment, as well as feeding sensory input from outside of the body into the central nervous system, and then also relaying the motor output. There's an awful lot going on. So our peripheral nervous system is actually broken down into several bits. So we can look at that on this diagram. So here we have the peripheral nervous system. So you can imagine that the central nervous system is over here doing its thing. We're looking specifically at the peripheral nervous system, and then we can see that it is divided into two separate systems. So we have the somatic nervous system, or the voluntary nervous system, if you like. So this is the part of this nervous system that we are consciously in control of. So if you want to bend down to pick something up, or you want to write something, or you want to brush your hair, or whatever it might be, this is stuff that you are controlling. So this is carried out by the somatic nervous system. But there is an awful lot of stuff going on that we have no conscious control of at all. And that is carried out by the autonomic nervous system. And the autonomic nervous system, again, because of its complexity, because of the range of things that it has to do, is itself divided into three separate divisions. So we have the sympathetic, the parasympathetic, and the enteric divisions. So the sympathetic division is associated with fight or flight. And I'll show you some of this on the next slide on a diagram. But you're all probably familiar with the fight or flight response. It's a defense mechanism that in a time of danger prepares our body for either running away as quickly as we can or standing and fighting for our lives. When we're not experiencing fight or flight, we should be within a range of rest and digest. So this is where our bodies are comfortable, they're relaxed, we're able to eat and digest food, and we're just going about our business. And when that's the case, we are under the control of the parasympathetic division of the autonomic nervous system. Okay, so sympathetic division is fight or flight, parasympathetic division, rest and digest. And when the parasympathetic division is in control, we also have the enteric division that is working alongside it to control our digestive tract or our digestive processes, basically. So the digestive tract, things like the pancreas and the gallbladder, so those secretory organs which are part of our digestive system, these are allowed to work when we're under the control of the parasympathetic division. When, however, the sympathetic division kicks in, it will override and basically turn off the enteric division. And we'll look at that in a sec. Okay, so these are the three divisions of the autonomic nervous system that we don't have any control of. So let's think about what actually happens when we're in a state of fight or flight. Now, unfortunately, this is, although we still do need it, obviously, because we do experience times of danger, but we've never lived safer lives than we do now. But in terms of our evolutionary past, it was really dangerous being alive in the world, and we would be exposed to predation from other animals. There would be the risk of war and fighting between groups of humans. So it was very, very important that we had this fight or flight response that prepared us for action. And what we can see here, if we look at this diagram, on the left, we can see the things that the parasympathetic division does. So this is the rest and digest. And here we've got the sympathetic division. So hopefully you're all under the control of the parasympathetic division at the moment. You're all quite calm. But let's imagine now that a lion came into your room. So now you're facing this very, very real danger. And your body will respond by triggering fight or flight. The sympathetic division of the autonomic nervous system will kick in. Here we can see some of the things that will happen. The pupils of the eye dilate. You start to switch off the digestive system. So you're moving blood away from the digestive system and you are focusing it on your muscles specifically, and getting as much glucose and oxygen to your muscles as possible so that you can generate loads of ATP to power muscle contractions. Okay, so that is the main thing with the sympathetic division with fight or flight. You want to get those muscles ready for action and keep them working. We stop producing saliva. I'll come back to some of these in a minute. The bronchi in the lungs will relax so we can get more air in and out more quickly. Our heart rate accelerates. The activity of the digestive system is inhibited, which I mentioned just now. So that includes the intestines and the stomach and the pancreas. What we get, though, is the stimulate glucose release from the liver. If you remember in previous sessions, we talked about the fact that glucose is stored up in something called glycogen within our muscles and our liver. Well, that glycogen during fight or flight can get broken down to start providing glucose to our muscles so they can go through cellular respiration to generate ATP for muscle contractions. So we start to break down that glycogen. We start to stimulate the adrenal medulla, which are endocrine glands that sit on top of our kidneys and produce adrenaline. We'll start to inhibit the emptying of the bladder. You might actually get a rapid release of urine or a rapid release of the emptying of the bladder. But once that happens, you will stop producing urine and filling and emptying the bladder. And one thing that you do see, interestingly, is that the one place where the parasympathetic and sympathetic divisions work together is within sex. So the sympathetic division promotes ejaculation and vaginal contractions, whereas the parasympathetic division actually promotes erection of genitals. So this is something where they work together. This is now preparing, all of these things are preparing your body for fighting for its life or just running away as quickly as possible. And like I said, this evolved over millions of years to keep us safe. But we today, as humans, live in a fairly safe world. There are still risks, but it's as safe as it's probably ever been. But this system still exists. So what we find is now, instead of being faced with a predator, this will now kick in when we are faced with an exam, or when we are faced with a driving test, or when we are faced with a first date, or doing a presentation, or something like that. Anything that is potentially frightening or potentially risky, our brains can identify as a high risk, as a high risk time or a high risk target. So that will then basically, the brain will try and jump to our defense by triggering the fight or flight response. But it's actually counterintuitive, because none of these things really are of any use to you when you're stood at the front of a group trying to or about to give a presentation, or you've just jumped into a car to do a driving test, or you're sat in a big room trying to do an exam. You don't want an accelerated heart rate and a dry mouth and tunnel vision that can come from your pupils dilating. Those things aren't particularly useful for you at that particular time. But you can actually train your body a little bit, or you can train your brain into good or bad habits a little bit. So if you, say for instance, have a presentation to do and you don't plan you don't plan properly for that presentation, and then you stand up to do the presentation and it goes really badly, well, your brain will pay attention to that. It will pay attention to the fact that you felt terrible. And then the next time a presentation comes around, your brain will go, I remember doing presentations. They're horrible and they make me feel bad. So I will protect us. And it will protect you by triggering the sympathetic division. Whereas clearly that's not good. And if you let it, it will take control of you every time you have to do a presentation, because it identifies them as something bad. But fortunately, you can train yourself out of that a little bit by actually doing lots of little things, so you can plan properly for your presentation and prepare and do lots of little things to help you through the way. So if you know you're going to get a dry mouth, have a glass of water available. If you know that you're likely to get tunnel vision and you're not going to be able to focus properly, if you're making notes, make them in a really nice big font so you can read them much more clearly. It can actually be really difficult to read small handwritten notes when you're trying to do a presentation, because the adrenaline's going, your heart rate's going, and your eyes open, your pupils dilate a little bit, so it's quite hard to focus. And then what you find is you'll do a presentation and it'll actually go okay. It'll be okay. And then the next time you come to do a presentation, it won't be quite as bad. You won't be quite as scared, and it will go a little bit better again. And before you know it, you get a few under your belt, and then you don't get scared anymore because you've taught your brain that they aren't a scary thing. Going back to our example then, so let's say, for instance, the lion has jumped into the room, the sympathetic division is kicked in the fight or flight response, and we have sprung into action. Let's not fight the lion because that probably wouldn't go down very well. So let's run. So we jump out the window and we run for our lives. Great. So we make it to a safe place. The lion's nowhere near us. And then we can start to relax. So the parasympathetic division will now start to take control of our bodies. So it basically reverses all of those things. So the eyes or the pupils will start to constrict again. So we can start to focus more clearly instead of just trying to get as much light in as possible so we can get a wider field of view. Well, now we can just focus on things more clearly. We can start to switch on the digestive system again. So we'll start producing saliva. Our breathing rate will start to reduce. So our bronchi will constrict or get narrower. Heart rate reduces. Like I said, the digestive system will start to switch on. So our enteric division will start to control these processes again. And then we can also obviously go to the toilet again. So everything should calm down and we'll return to a state of fight or flight, a state of rest and digest again. So those are the parasympathetic and sympathetic divisions of the autonomic nervous system. The next time that you find yourself in a position, if you do experience anxiety or panic attacks, which are basically the fight or flight response, if you find yourself experiencing them when you don't want to, try and keep in mind that this is my brain's way of trying to protect me, which is great. It's your brain trying to look after you. But if it's in a place where you don't want it to be there, you can take steps to try to teach your brain not to do that in certain circumstances. So it's not an easy process to go through, but it's ultimately worth it if you can do those challenging things like presentations and exams and driving tests and dates or whatever it is that can trigger these things. Obviously, life can be a lot simpler if you don't experience them. So anyway, moving on. Now that we've set the scene of what nervous systems are and a little bit about what they actually do, the next thing we need to do is go on to talk about how they do what they do. And we can start off by looking at the structure of the main component of nervous systems, which are the neurons. And we're going to focus specifically on vertebrate neurons. And as we go through, I'll explain why invertebrate neurons are a little bit different. What we can see on the diagram here are actually two neurons. So let's start up here on this left-hand neuron here, and we can identify and talk about some of the key components of it. So let's start up here. Then most neurons have dendrites, which are highly branched extensions that receive signals from other neurons. So these bits that look like branches or roots, if you prefer, coming out of the neuron up here, these are called dendrites. And these essentially act like attachment points for other neurons that are feeding into this particular neuron. The next bit that we see down here, this is the cell body, and this is the main typical part of the cell that you would recognize. So it has the nucleus and the endoplasmic reticulum and the Golgi body and all of those kinds of things in there. So you have the cell body up here, and then the cell body will start to narrow, and we call that bit the axon hillock. There's some good names within the structures of neurons. So we narrow down into the axon hillock, and then we have the axon. Okay, so the axon is typically a much longer extension that transmits signals to other cells. So the axon is this long bit that looks a bit like a tail, and it is this part of the neuron that is sending the nerve impulse, or what we correctly call, or properly call, a action potential. So an action potential is the correct term for a nerve impulse, and the action potential starts here at the axon hillock, and then it will travel along the axon down to what we call a synapse. So a synapse is a connection between a neuron and a postsynaptic cell. So there's going to be a lot of these terms flying about over the next few slides. So let's try and break them down a little bit here. So information is transmitted from a presynaptic cell. So basically a synapse is a junction between two two nerve cells, essentially, or between a nerve cell and an effector cell. So in this instance, the synapse is going to be down here. So you can see the synapse in that little square there. We come down to the end of our axon and we have the synaptic terminals. So each synaptic terminal will be able to form its own synapse. So we're just going to take this synapse up here, which is in this box. So when we talk about the presynaptic neuron, what we're talking about is this neuron, because it comes before this synapse. And then when we're referring to the cell that's after the synapse, we would call that the postsynaptic cell. In this case, it's another neuron. So we can call that the postsynaptic cell or the postsynaptic neuron. It doesn't really matter. The key bit is there is the synapse. So the neuron before that, that's feeding the information into the synapse. Well, this is the presynaptic neuron. And because that this neuron here is after the neuron, after the synapse, this is the postsynaptic cell or the postsynaptic neuron. And the reason that we know this, the reason that we know that if this is our synapse, this is the pre and this is the post, is because information only travels along an axon in one direction, and it will travel from the cell body to the synaptic terminals. So it will only travel in this direction. Okay, so if you've got a synapse here, you know that this is the presynaptic neuron and this is the postsynaptic cell. This could be a neuron like it is here, but it could also be a muscle cell. So this neuron here, the presynaptic neuron, could be sending a message for this, if it was a muscle cell, for this muscle cell to contract. Okay, so that's how this could work. It could be a gland cell, so an endocrine gland. So this neuron could be sending an impulse or an action potential to an endocrine gland to secrete a specific hormone. Okay, so we're going to have a bit of a closer look at neurons in a moment. But I just wanted to get across the key structures within neurons. So we have the cell body, and then we have the dendrites coming off of the cell body. We then narrow down into the axon hillock, and then we have the axon itself, and then the axon terminates at the synaptic terminals, and those synaptic terminals can form synapses with other cells. And those other cells, they could be other neurons, they can be muscle cells, they can be endocrine glands. These other bits so that I haven't mentioned yet are the myelin sheath. So these are made up of glial cells. So these are supporting cells, and these help to insulate the axon, and they will actually speed up the action potential as it moves along the axon. And I'll talk about how it does that in a bit. Okay, so that's the basic structure of a vertebrate neuron then. Cell body, dendrites, axon, synaptic terminals forming synapses with a postsynaptic cell. And then, as I've already mentioned, you've also got the myelin sheath that wraps around the axon to speed up the rate of transmission of the action potential. But now that we know about the structure of a neuron, we need to talk about the action potentials. This might be one of those things one of these little bits that you need to go over a couple of times. So I'm going to break it up with an animation that will, or a video from a YouTube presenter, which might help as well to go over it a couple extra times. Action potentials then, these are signals that carry information along axons. So if you think about what, at its crudest point, a nervous system is trying to get information from point A to point B. Okay, so if you can imagine you've got light entering your eye, you need to get that information from your eye to your brain. And then you need to get information from your brain to the muscles in your legs. Or you need to get information from the tips of your fingers where you feel something to your brain, and then another message from your brain somewhere else. So you're sending information from point A to point B. So how do you do that? How are you, on a very simple biological level, going to transmit a signal or transmit information from point A to point B? Well, if you think about it, one of the easiest ways of getting things spread over a wide distance would be diffusion. So we've talked about diffusion before. It's how we get things like oxygen from our lungs into our blood, and from our blood into the inside of our cells. We know that it's the movement of substances from an area of high concentration to an area of low concentration until they are evenly distributed. So in theory, you could then have a neuron as a hollow cell, and then you release substances into one part of the cell, and then they will diffuse throughout the cell, sending their message. The problem with diffusion is it can be quite a slow process. It doesn't necessarily happen that quick. And when you get into larger animals, you're trying to send information over quite a long distance. So simple diffusion doesn't really work over those long distances. If you see a lion coming towards you, wanting to eat you, you don't have 10 minutes to wait for a signal to diffuse passively throughout your body, because you'll be dead, you'll be eaten. So we need our nervous systems, if they're going to be useful, they need to happen incredibly quickly. Instead, what we actually do is we do rely on a type of diffusion, but instead of doing this over our entire length of our neurons, we do it over very, very small sections of the membrane of our axons. Action potentials are caused by changes in electrical charge across the plasma membrane of axons. So specifically, what we are doing is changing or measuring, when we look at these, we are measuring the changes in electrical charge caused by the diffusion of sodium ions and potassium ions through special ion channels within the membrane. And because of this, because we're doing it just over small sections of the membrane, a single neuron can produce hundreds of action potentials per second. And as we mentioned, let's get rid of that bit for a second. I'll come back to that. So let's have a look at using this diagram very simply what I mean by how action potentials work through diffusion across small sections of membrane. So we'll start up here. We can see an axon here, or a neuron here. We're having a close-up of an axon. This one is showing an unmyelinated axon, just so it's easy. So we're looking at a section of axon here. So this is what we can represent here. So when a neuron isn't firing, when it's not sending an impulse or an action potential, it is in what we refer to as the resting state. And when our neurons are in their resting state, we have sodium-potassium pumps, which is a special protein within our membranes that will actively transport. So if you remember, that requires the use of ATP energy. So they will pump sodium ions out of the cell. So we can see the inside of the cell here. They will pump sodium ions out of the cell, and then they will pump potassium ions into the cell. And what that does is it creates concentration gradients. So we end up, when our cells are in the resting state, we will end up with an abundance of sodium at just outside of the membrane of the axon, and we will end up with lots of potassium on the inside of the cell. And they basically, just like anything else, they want to travel down their concentration gradient. So potassium wants to flood out of the cell, and sodium wants to flood into the cell. But they can't. There's no way that they can get in when the cell isn't firing. Instead, what will happen is when we get a stimulus that comes into the cell body, or the dendrites, what that will do is it will trigger something called a depolarization. And depolarization will cause specific channels that are called voltage-gated ion channels. Okay, so they will open in response to a stimulus, and they will specifically let sodium rush into the cell. So all of the sodium that's built up on the outside here because of the sodium-potassium pump, because of that stimulus, will now flood into the cell. And that changes the electrical charge across this membrane. So you can see here, it's now, because sodium is positively charged and the potassium ions are positively charged, well, also in here, you've got lots of negatively charged chloride ions, you've got negatively charged proteins. Well, now that you've got positively charged sodium flooding in, that makes the inside of this part of the axon very, very positive in relation to the outside of the cell. So you can see that it's positive in here, negative in here. What that does then is it changes the voltage across the membrane, and that will trigger, or it will act as a stimulus to trigger opening of ion channels or sodium channels in the next part of the axon. So this process of opening up sodium channels, sodium rushing in and making the inside of the axon more positive, is something that we call depolarization. So depolarization of this part of the axon will trigger depolarization in this part of the axon. And that will then trigger depolarization in this part of the axon, which will trigger depolarization in this part of the axon, and so on and so forth. That is how you get from your cell body all the way down the axon to the synaptic terminals. You depolarize a part of the cell. It typically starts here at the axon hillock, the action potential. So you'll get depolarization up here, which depolarizes the next bit, which depolarizes the next bit, and so on. It's very much like having a row of dominoes lined up. If you hit that first domino hard enough, you will trigger that domino to fall over, which hits the next one, which hits the next one, which hits the next one. So you will end up with all of them falling over. And that is how an action potential works. It is an all or nothing response. When you get a stimulus that is strong enough to knock over that first domino, so a stimulus that is strong enough to open up those first sodium channels, that will cause depolarization, and then your action potential is happening whether you want it to or not. It will depolarize the next bit, the next bit, the next bit, the next bit, and it will go all the way down to the synaptic terminals. But if you remember, it's very important that, or it's an important component of neurons, that they only transmit an action potential in one direction. So they only go from cell body down to the synaptic terminals. What you don't want is an action potential traveling back, because otherwise you can just end up with a nerve impulse getting stuck within the axon and bouncing back and forth. Because you can imagine that if this bit depolarizes this bit, well, there's nothing stopping it then from depolarizing this bit, so it can travel backwards. And that's no good. You want your action potential traveling rapidly in one direction. So what will happen is once you've depolarized this part of the axon and then it shifts onto this bit will then go through a process called repolarization, and then it will move into something called hyperpolarization. So you can see here that the potassium channels have now opened, and potassium is flooding out of the cell. And then what that does is it causes the inside of the cell to be more negative, because the potassium channels are taking positive charge with them. What that does, it takes the membrane potential away from something that we call the threshold, and the threshold is the point at which the action potential will actually be triggered. So this is an action potential. It starts typically up here at the axon hillock. We are depolarizing, so opening up sodium channels to allow sodium to rush in. That will then cause depolarization in the next bit of the axon, and the next bit of the axon. And then once a bit of axon has been depolarized, that will then trigger the opening of the potassium channels, causing the potassium to rush out of the cell, making the inside of the cell more negative, ensuring that the action potential will not travel backwards up the axon. So they're only going to travel in one direction.
Hello and welcome to Interactive Biology TV, where we're making biology fun. My name is Leslie Samuel. This is episode 13, where we're going to be talking about the action potential. I'm basically going to be giving you a review of the concepts that we've been talking about up to this point when it comes to the action potential. Over here, we have our neuron, and the part of the neuron that we are going to be focusing on is the axon. So that's this region here, starts here, and it goes to about there. Now, what I'm going to do is I'm going to take a section of the neuron. So let's say I'm going to take this part here, and I am going to draw it down here. So here we have the axon. This is inside the axon, and this is outside the axon. Now, if you remember from a previous episode, outside the axon we have a lot of sodium ions. So I'm going to draw sodium ions here, and they are all outside the cell. Now let's look over here. Here we have a stimulus that's happening, and you can see there's a first stimulus that does not reach threshold, so nothing happens. Another stimulus comes, it does not reach threshold, so we still do not get an action potential. If we have a stimulus that's strong enough, and I'm going to draw another stimulus in here. So let's say we have a stimulus that's that big. That's going to reach the threshold and cause an action potential. When that happens, voltage-gated sodium channels are going to open, and that's going to cause sodium to rush into the cell. Of course, that's going to start at the axon hillock. Sodium is going to rush in, making the membrane potential even more positive, causing more channels to open, more sodium to rush in, more sodium to rush in along the axon. What is that going to do to the membrane potential? You can see right here, this is where we get depolarization. The membrane potential goes up and up and up and up, and it's trying to reach the Donan equilibrium for sodium ions. And that equilibrium potential is somewhere around 58 millivolts. So sodium is rushing in because of the driving force causing sodium to go in. Sodium wants the membrane potential to go up to its Donan equilibrium, and that is around 58 millivolts. At this point, voltage-gated potassium channels have enough voltage in order for them to open. So I'm going to erase all of this now, and we're going to take a look at the axon again. I'm going to draw it here in blue. And inside the cell, we have a bunch of potassium ions. Now we have such a positive charge on the inside that potassium ions want to leave because positive repels positive. When voltage-gated potassium channels open, potassium can now leave the cell. So we have a lot of positive leaving the cell. And what that is going to do is cause repolarization, where the membrane potential is going down. Just like with sodium, potassium wants to reach its equilibrium potential, which is somewhere around negative 93 millivolts. And that is why the membrane potential is going down as it's leaving, because potassium wants the membrane potential to be at negative 93 millivolts. That is where it is most comfortable. Considering that the resting membrane potential is around negative 70, the membrane potential goes significantly lower than that negative 70, and this phase we call hyperpolarization. All along this process, we have the sodium-potassium pumps that are pumping sodium out of the cell, 3 sodium ions out, and 2 potassium ions back in. And that's going to cause the membrane potential to eventually reach back to its resting state. This is the entire action potential, looking at depolarization, repolarization, hyperpolarization, and it getting back to the resting state. I hope that makes sense. If you have any questions, feel free to leave them in the comments below. I'd be happy to answer your questions and maybe even make a video answering your specific question. That's it for this video, and I'll see you in the next one.
Okay, so hopefully that was useful. As I said, action potentials are a little bit of a tricky topic to get your head around when you first look at them. So it's worth just going over them a few times, particularly if you've got a good textbook, sort of do some reading on it, and eventually it will definitely click into place. It's certainly nothing to be afraid of, just takes a bit of going over, that's all. what we just looked at then is what action potentials are and how they actually work. But how does that tie in with the glial cells, specifically the myelin sheath that we see around the axons of our neurons? Well, they actually work in combination with each other. So what we know is that when you look at an axon, because we are essentially looking at the flow of sodium and potassium ions in and out of the neuron, that the speed of an action potential will increase with the axon's diameter. So I always think of it a bit like water going through a hose pipe. The wider the hose pipe, or the wider the diameter of the hose pipe, the more water you can get through that hose pipe more quickly. And so it is with the axon. The wider the diameter of the axon, the faster an action potential can travel along it. The problem comes with the fact that for an animal such as us, we have very, very complex brains, and our brains are made up of billions of neurons. So if we had to rely on having very wide diameters, we would either have to be very, very simple in terms of having very simple brains, or we would have to have massive heads and massive brains to allow for all of those wide diameter axon neurons. Instead, what we've done is we've managed to maintain the speed of our neurons whilst also having very narrow neurons or axons. And we do that through insulation with the myelin sheath. In vertebrates, like ourselves, axons are insulated by this myelin sheath, and this causes an action potential speed to increase dramatically. If you look at an invertebrate, such as squid, for instance, that also have very fast-acting nervous systems, well, they've gone down the different route. So they have unmyelinated axons, and for their nervous systems to work quickly, well, they've had to go down the route of having large diameter axons. So they're often referred to as giant axons, and they do work very, very quickly, but they're some of the largest cells within the animal kingdom, and although it doesn't sound massive, they can get up to 1 millimeter in diameter, which for an individual cell is pretty big. So let's have a look at this diagram here. You can see that you've got the myelin sheath around the axon, but if you notice, it doesn't cover the whole of the axon. There are bits of axon exposed, and we call those bits of exposed axon the nodes of Ranvier. So we can see a close up here. You can see an exposed bit of axon here called the node of Ranvier. Here you can see how the myelin sheath, which is a fatty layer, actually wraps around the axon of the neuron. And essentially what will happen is that when the action potential is generated, so depolarization occurs up here at the axon hillock, what will happen is you won't get depolarization here because it's covered in the myelin sheath. Instead, what will happen is depolarization occurs at the axon hillock, and then it skips to depolarizing the first node of Ranvier, and then it will skip to the next node of Ranvier, and then it will skip to the next node of Ranvier, and so on and so forth. So instead of having to go through depolarization of all of the bits of the axon, instead what will happen is it will skip from node of Ranvier to node of Ranvier. And that dramatically increases the speed at which an action potential travels along the axon. And we actually call that process saltatory conduction. So we know that in vertebrates, axons are insulated by the myelin sheath that causes the action potential speed to increase. So action potentials are formed only at the nodes of Ranvier. So those are the gaps in the myelin sheath where we have our voltage-gated sodium channels. So action potentials in myelinated axons jump between the nodes of Ranvier in that process called saltatory conduction. So you can see it here with the red arrows. So this is why our axons can transmit action potentials very quickly while still maintaining a very narrow diameter. And there are certain diseases that we can get that damage, or we can damage the myelin sheath as we get old, but you can also get certain diseases that will actually damage the myelin sheath, and that means that the nervous system doesn't function as well as it should because you're not getting saltatory conduction happening effectively. So what was the whole point of this process of action potentials then? Well, it's, as I mentioned right at the beginning of this, it's to transmit a signal from point A to point B. So when we're looking at an individual neuron, point A will be the cell body or the dendrite, and point B will be the synaptic terminal. But like I said, we can do this over a longer distance and say it's going from the tips of our fingers to our brain, and then we're just traveling along multiple neurons. But we're still just about moving a message from point A to point B. So what actually happens when we get that message to point B? Okay, so what actually happens when we get that message to the synapse? What's actually going on? Well, that's what we need to look at next. A synapse is a junction between an axon and another cell. We established that earlier on. So the pre-synaptic neuron is delivering information to the synapse, and then you'll have the post-synaptic cell, which will be another neuron. It will be a muscle cell or an endocrine gland, typically. And interestingly, if we look here, so we can see here a synaptic terminal of a presynaptic neuron, and then we can see the membrane of the postsynaptic cell. Well, they don't touch each other. There's no physical connection between them at a chemical synapse. Instead, what happens is you have a gap between them, which is called the synaptic cleft. And instead, what happens is the presynaptic neuron releases neurotransmitters into the cleft, which will then diffuse across the cleft and then attach to receptors within the membrane of the postsynaptic cell. Okay, so that's typically, and we'll look at this in a bit more detail on the next slide. So a synaptic terminal of one axon passes information across the synapse in the form of chemical messengers called neurotransmitters. So I'm sure you're all familiar with neurotransmitters. It's some of the more famous ones like serotonin and dopamine. But there are lots of them within the human body. So information is transmitted from a presynaptic neuron to a postsynaptic cell. Well, we've established that already. So we get two main types of synapse. So electrical synapse, what we're getting is basically just the continuous flow of the electrical current. So essentially the action potential just simply passes from the presynaptic neuron to the postsynaptic neuron. Okay, so these are fairly rare though. Most of our synapses are chemical synapses. And at chemical synapses, a chemical neurotransmitter carries information across the synaptic cleft. So that's exactly what we can see here. And what we can see now is the whole point of the action potential. So what will happen is, here we can see our synaptic terminal of our presynaptic neuron. Here we can see our postsynaptic cell. Here are those receptor proteins embedded within the plasma membrane of our postsynaptic cell. And here we can see the synaptic cleft, which is the gap between the two cells. Inside of our synaptic terminals, we have these vesicles that are little just bags of membrane that contain neurotransmitters. And they are just sort of within the synaptic terminals. Within the membrane of our synaptic terminals, we have these voltage-gated calcium channels. Okay, so these will open in response to a certain change in electrical voltage. For instance, an action potential. So this is exactly what happens. An action potential, so depolarization, will travel from the cell body all the way down here to the synaptic terminals. What that does is when it causes depolarization of the synaptic terminal, it causes these voltage-gated calcium channels to open. Calcium will then start to flood into the synaptic terminals. This calcium will then cause these vesicles to embed within the plasma membrane here, releasing the neurotransmitters into the synaptic cleft. The neurotransmitters will then diffuse across the cleft where they bind to the receptor proteins, triggering a specific response within the receptor proteins. Now those receptor proteins may open and allow sodium to rush in, which will cause depolarization within this if it's a postsynaptic neuron. It might be that the neurotransmitters trigger potassium channels to open, which will cause potassium to flood out, which will cause hyperpolarization. Okay, so a lot of different things can happen at these synapses depending on what neurotransmitter you have, depending on what receptor protein you have, and depending on what the postsynaptic cell is. So you can get lots of different responses. What will happen then is once the action potential has happened, the neurotransmitters have been released and received, and the postsynaptic cell has responded. What will happen then is you've got these neurotransmitters that have to be dealt with. You need to get rid of them because otherwise these postsynaptic cells will keep responding to them. And typically they will be gotten rid of in one of two ways. So the first way is that there will often be enzymes within the synaptic cleft that will break down the neurotransmitters into their constituent parts that will then get fed into other processes. So a good example of that would be in something like, we actually talked about this as an example when we looked at enzymes, and that is Viagra. So Viagra actually works by inhibiting an enzyme that breaks down the neurotransmitter nitric oxide. Okay, so you can imagine that nitric oxide is one of these little green balls here, the neurotransmitter that will trigger the dilation of muscle tissue within the penis. And then the enzyme will break down that nitric oxide, causing constriction of the blood vessels. Well, the enzyme will basically, or the Viagra will inhibit that enzyme. The other thing that can happen is the presynaptic cell, so in this case here we've got the synaptic terminals, will take up the neurotransmitters. They will reuptake them, if you like, and package them back up into vesicles so they can get used again. And this is something that we see with a very common neurotransmitter called serotonin. And there's a type of drugs that are commonly used called SSRIs, or selective serotonin reuptake inhibitors, which is a type of antidepressant. And they essentially work by preventing the reuptake of serotonin, which means that the serotonin stays within the synaptic cleft for longer, triggering more responses in the postsynaptic cells, which means that serotonin gets to work for longer. And what serotonin does, and one of the things that it does within the brain, is it creates a sense of well-being and calm. So that is basically how synapses work. Okay, so that's nervous system. So we've talked a little bit about the different types of nervous systems, and we've looked at how they actually function. So we've looked at the structure of individual neurons, and we've talked a bit about action potentials and how they work. Next, we need to finish off by having a look at different types of sensory systems in animals and what they specifically do. Now, this is going to be very, very dependent on which species of animal you're looking at, because we see a huge amount of diversity in specific methods in which animals sense the environment around them, but they can come under a few main types. So those are what we're going to focus on today. So sensory systems convey information about an animal's environment to its brain, and then muscles and skeletons carry out movements as instructed by the brain. That's kind of what we've established as we've gone through. You see the star-faced mole here with its highly sensitive nose that it uses for detecting the environment and ideally finding food, such as earthworms. So sensory receptors detect stimulus energy and transmit signals to the central nervous system. So basically all stimuli represent forms of energy. And a sensory receptor is able to convert that energy into a change in the membrane potential. So it's the energy within a particular stimulus that will ultimately trigger an action potential. So when a stimulus is received and processed by the nervous system, a motor response may then be generated. And when we get to that point, it may be a simple reflex reaction, or it may be a much more elaborate thing that's going on. So you might be familiar with a reflex action or a reflex arc. These are very, very simple reactions that can often occur within the spinal cord, and they don't have to go all the way to the brain. And this saves valuable time. So a classic example of this would be if you accidentally touch your hand onto a hot saucepan or a hot kettle. You instantly remove your hand from that kettle because that information has gone from your hand to your spinal cord. It hasn't had to go all the way to the brain. There's a certain amount of processing or integration that can go on within your spinal cord. If you need more elaborate processing, well, that will then need to go to the brain for further integration. So what are the types of sensory receptors that we get then? Well, there are 5 main categories, and we're going to go through these one by one. So there are mechanoreceptors, chemoreceptors, electromagnetic receptors, thermoreceptors, and pain receptors. And between them, they cover our 7 main sensory systems. So we obviously have visual for sight, auditory for hearing, gustatory refers to taste, smell is olfactory, touch is tactile, vestibular is what enables balance, and then the proprioception, which is body awareness. So let's go through the 5 main types of receptor then. So mechanoreceptors sense physical deformation caused by forms of mechanical energy. So they typically consist of ion channels linked to structures that end outside the cell. So we're talking about things that are built into our integumentary system, such as our hairs. So the mammalian sense of touch, as an example, relies on mechanoreceptors that are dendrites of sensory neurons. So if we look at this diagram of our skin, what we can see is within the hypodermis here. Remember, we've looked at this before. So you've got the hypodermis, the dermis, and then the epidermis. Well, we've got nerves that come in through the hypodermis, and then you've got these dendrites, these extensions within the cell that move up into the dermis and then also up into the epidermis. So these will then respond to pressure and vibration. The next type of receptor that we've got are chemoreceptors. So these transmit information about the total solute concentration of a solution, but other chemoreceptors will actually respond to individual kinds of molecules. So there's been some interesting information on this. Actually, I'll mention that in a second. When a stimulus molecule binds to a chemoreceptor, the chemoreceptor becomes more or less permeable to ions. So this, for a long time, and even today really, it's accepted that molecules and the receptors work a bit like a lock and key. So the scent molecule is a key and our chemoreceptor is a lock. So a molecule will come along, it'll be the right shape and size to bind to the chemoreceptor, which will then trigger the response. But there's been some quite interesting work done in an area called quantum biology that suggests that rather than working as a lock and key hypothesis or on a lock and key basis, that it's more down to the vibration of the molecules, because this would be more likely to explain why molecules that have very, very different size and shape, for instance, would trigger the same response in us. So why 2 things that are completely unrelated might smell the same. So the classic example would be something like cyanide, which is often reported as smelling like almonds. And the scent molecules within almonds are very, very different to the molecules of cyanide. So they shouldn't, in theory, smell anything like each other. But this is fairly new science, and it's not 100% certain at the moment, but there's some pretty compelling evidence to suggest that it might be down to, on a quantum level anyway, looking at the vibrations within those molecules. And if that's something that interests you, I would certainly recommend that you look into it in a little bit more detail. So different animals obviously will rely more or less heavily on their sense of smell, or relying on chemoreceptors depending on in sort of what environment they live. So as an example here, we have the antennae of male silkworm moths have very sensitive specific chemoreceptors. So we can see the moth here, and you can see the chemoreceptors on its antennae. And they are basically very sensitive to scent molecules that are given off by the females. So it helps the males to find females to help them reproduce. Okay, the next one is electromagnetic receptors, and these detect electromagnetic energy such as light, electricity, and magnetism. And there are lots of interesting animals that use electromagnetic receptors. So an example here would be the platypus. So it has electroreceptors on its bill that can detect the electric field generated by prey. Many animals will also use electric or electromagnetic receptors in the brain to be able to respond to the Earth's magnetic field, and this will help them to migrate. So it's suggested that certain butterflies, like monarch butterflies that undergo mass migrations or long migrations, but also certain marine mammals. So here we can see an example of beluga whales, which are believed to use this process to enable them to migrate. Monarch butterfly, which I mentioned just now. But here we can see these pits that are located within the rostrum or the nose of this shark. Well, these are called the Ampullae of Lorenzini. And when you look at them in a bit more detail, what you can see is you have a pore, which is these dark spots that you can see here. And these pores open up into these canals that are filled with an electrical conductive gel. And this transmits electrical current down to the ampulla, which are essentially sensitive hairs almost. And they basically then transmit this signal via nerves to the brain. And this enables sharks to detect changes in electrical current created by things like muscle contractions within their prey animals. The next type then are thermoreceptors, and these, as you'd imagine, with a name like that, detect heat and cold. So we go from the basic ones that we have, that we experience on a day-to-day basis when we can touch something and sense whether it's hot and cold, to sort of more high-tech ones like we see in snakes, for instance, that are able to rely on thermoreceptors to detect infrared radiation emitted by prey when they're obviously warm-bodied prey. One example where this can actually be tricked is with peppers. So when you look at the chili peppers, like jalapenos and cayenne peppers, they contain a substance called capsicum. And this is actually a molecule that will bind to receptors within our mouths that detect heat. It's how we respond to hot substances. Like if we drink a hot cup of coffee and that's too hot, it's essentially what enables us to know that we need to not consume any more of that. It's not that the chili pepper is actually hot or it's causing the physical damage associated with drinking hot coffee, for instance, but it basically tricks our brain into thinking that it's very, very hot. And our body will respond accordingly. And it does this by opening calcium channels. And as we saw just now, calcium channels opening in the synaptic terminals are what enable the transmission of impulses or action potentials at our synapses. So we have, as mammals, we have a variety of thermoreceptors, and each specific for a particular temperature range. So these can be quite complex. So here are the heat sensing organs within this viper, and then obviously lovely chilies. Based on that, we really shouldn't like the heat that comes from chilies, but clearly a lot of people really do. And there's some interesting research going into that. So if you're a big fan of chilies and you like chili heat and you don't understand why, it's worth having a read into. There's all sorts of benefits that come from eating chilies actually. The last of our type of receptors then are the pain receptors or nociceptors, and these detect stimuli that reflect harmful conditions. So they respond to things like excess heat, but also excess pressure or noxious chemicals released from damaged or inflamed tissues. So chemicals produced in an animal's body sometimes enhance the perception of pain. So there could be certain chemicals that are produced when we physically damage our body or we get a sort of blunt force trauma that can actually increase the perception of pain. Okay, so let's do a quick recap as always. So this session has been a little bit long so far, so we'll do this and then we'll bring it to an end. So as always, I'll read through the questions and then I'll give you 5 minutes to answer them, and then I'll go through the answers with you. So question number 1, what group of animals have simple nerve nets? Number 2, what is cephalization? Number 3, put these in the correct order. So you've got integration, motor output, and sensory input. So in what order should they be? And then number 4, why is the myelin sheath important? And then number 5, what do nociceptors sense? Okay, so I'll give you 5 minutes to answer those, and then we'll go through the answers together. Okay, welcome back. Hopefully you've had a chance to go through and answer all of those. So number 1 then, what group of animals have simple nerve nets? Well, the answer is the Cnidarians. So we're looking at things like jellyfish, corals, anemones, and the Hydra that was on our diagram earlier on. Number 2, what is cephalization? Well, it's the clustering of sensory organs at the front of the body or around the head. Number 3, put these in the correct order. Well, it should be sensory input, integration, and then motor output. And then number 4, why is the myelin sheath important? It insulates axons and increases the speed of action potentials through that process called saltatory conduction. So you can give yourself a bonus point if you wrote saltatory conduction. And then number 5, what do nociceptors sense? It's pain. Okay, so hopefully you're not feeling too much of that after this session. I know that we've covered quite a bit, but hopefully that was useful. So we've looked at a brief introduction to the diversity in nervous system. So remember, the important thing there, structure and function. So the more complex an animal's life, the more complex its nervous system. Okay, we then went on to focus on the vertebrate nervous system. So looking at the central and the peripheral, and then looking at the sympathetic, parasympathetic, and enteric divisions of the autonomic nervous system. We then went on to talk about action potentials and what they are, and finished off that section by talking about synapses. And then we just finished off by mentioning the main sensory systems within animals. Now, obviously, as I've gone through, I've pointed out that particularly the sensory systems, they're really diverse within animals. So depending on what type of animal you're looking at, it's worth looking specifically at those and getting a good feel for what systems they have, because it's a very diverse area. But anyway, as an introduction, I hope that was useful, and I'll see you in the next session.